Thank you so much, Professor Shipia, for hosting me and inviting me to University of Tokyo. So I'll be talking about roadmap for fast and efficient genome analysis. And before that, please let me introduce my research group again. So we are called Safari Research Group, and this is our motto: think big, aim high. And we have a large number of PhD students, masters, interns, and administrators, as well as postdoc and lecturers. So we are located in Switzerland, in the beautiful city of Zurich, and this is the city center, this is ETH. And we are part of computer science department. So our research group is led by Professor Onur Mutlu, who was a professor at CMU for 7 years, then he moved in September 2015 to ETH, Switzerland. And our research spans everything from computer architecture to hardware-software cooperation, hardware security, bioinformatics as well. And we are hiring students. We have openings at all levels: internship, master's, PhD, postdocs, and even visiting professors. So please feel free to contact me or Professor Onur Mutlu or apply directly to our application system. Okay, this is a personal story where we got these packages, contain some really beautiful things, including personal pictures of Japanese family. And they send these packages through JICA to Gaza City or Palestine. And we used to receive these as award for the best 2 or 3 students in each class in the school. So I was lucky enough to receive one of these, and I really appreciate it 15 years ago. And I think it's still moving on, still continue this nice thing until now. And I'm really proud of these, and I'm still keeping them in my parent house. So I would like to thank all Japanese people for supporting education. Thank you so much. All right, so now we move to the talk. So I'm not going to talk about with lab process. I think you are smart enough to know all these things more than I do. But what I would like to talk about for this today talk is how to design the algorithm or computer architecture or accelerator to accelerate some of these processes in genome analysis. So I will talk first why we pick genome analysis as a problem to be accelerated and what is actually genome analysis. And then I will jump in how we do read mapping as a basic step, and then the algorithm and the accelerator that we use to accelerate this step. And then I will show you some future opportunity to do even further and accelerate the process much faster. Okay, so why genome analysis? So most of you know this term. And this term means difficult illness, which is a term coined by the Japanese Ministry of Labor, Health, and Welfare that refers to a rare disease. And I think difficult illness is more accurate than rare disease because it's no longer rare. So based on the definition, some people define it as 5 in every 10,000 people who get this rare disease. But it's also expected that every 17 people that they have one of them developing or having a rare disease at different stage of their life. That is translating to number to 350 million people in the worldwide. And 75% of these people are children. And 30% of these children never make it to their 5th birthday. And 80% of these rare disease or difficult illness are caused by genetic disorder. So Professor Matsuda from Kyoto University said, we don't know exactly how many people in Japan have a rare disease, but you can think about it as 20 of your friends, one of them will have a rare disease. So you can think about the importance of having a personalized medicine, for example, to address these rare disease, or the need for reading their genome such that we know what genetic variation they have or what caused such disease. So Japan has an initiative on rare diseases where they spend around 600 to 700 million Japanese yen every year to read the genome of those patients with rare diseases. So they are able to sequence the genome of 2,000 patients a year with that cost. And in the UK as well, this year, they start doing whole genome sequencing for all critically sick children that hospitalized in the UK. And by doing so, and knowing what SNPs they might have, so from the phenotype or the symptoms, they might not be able to recognize what disease they have. So by doing whole genome sequencing, they might correlate this relationship between the patient to a certain or a previous case that they had. And by doing so, they are saving the inpatient cost by $100,000 to $300,000 per patient. So you can think about the importance of applying the same method in Japan, for example, or other countries. So here's an example for blood pressure. So this is a toy example. And for each row here, we have a portion, small portion of individual genome. So each row is a separate genome. And we have SNP1, for example, and SNP2. And because you are smart people, you can recognize that SNP2 has nothing to do with blood pressure because it seems like random values, right? And for SNP1, whenever you have a C at this location, then probably you might have a high blood pressure. So you can think, will we have only a single SNP that causes such diseases? Of course no. It's a bit more challenging, more complicated than this. So in these phenotypes, for example, if you delete a 500,000 character from your genome, you might get one of these two diseases, autism or obesity. And if you duplicate the same amount in the same chromosome, which is chromosome 16, you might get the opposite diseases. For example, obesity, you will get the underweight. And all of these observations are published in this paper. So I have the citation here. So it's really challenging to know what causes such disease. It might be single variation, or it might be very large structural variation, for example, insertions, deletion with the large portions. And the other application of reading our genomes, you can think about city-scale microbiome profiling, where you can go to very crowded places, such as Tokyo subway station, for example, or New York subways, and you would like to know what kind of species you have in this train. And this is a project published in 2015, where they go to New York subway station and swap about 1,500 samples in different locations, starting from the train seats, train door, train machine, the card machine, and everything related to train stations. And then they do some bioinformatics analysis, where they do read mapping to existing references and so on. And by doing that, they found that around 50% of these samples having bacteria. Some of them are useful. And the other 50% of these species exist in these samples are unknown. They couldn't have any references or previously sequenced that they could correlate it. So the most interesting thing here, they have black in their samples. And this caused by Yersinia pestis bacteria. And for those who don't know this caused in the 14th century, killed more than 1/3 of the population of Europe within a few years. So it's really dangerous to find such bacteria in New York subway station or anywhere in the world. And this raises some alarm in the media in that time. However, when they further investigated, they find it was a false alarm, false positive. Yersinia pestis wasn't there. And some people calling this as a failure of bioinformatics. And I totally disagree with this, because bioinformatics doing really good. So it's just a matter of choice. Which tool did you use to do the read mapping, for example, or to do the analysis of these samples compared to the reference gene? So that shows you the critical need for fast, and not only fast, but also accurate genome analysis. We need to not compromise the accuracy of doing genome analysis. Okay, so we show you the importance of doing genome analysis in a fast manner for critically ill children, for example, and also it should be accurate. Now, what is genome analysis? And I'm really sure that all of you are aware about these topics. So to do genome analysis, you need the genetic sample and convert it to the full text of our DNA, such that you can compare the text to another text using some computer algorithm rather than with lab methods. Unfortunately, we don't have any machine that can read our DNA. What we do have is a machine that can read portion of our DNA, very few segments or small segment. This segment could be very short or very long, but we cannot read the entire content of our DNA. So how long the DNA? So for viruses, a few thousand, for bacteria, a few million, for a human being, a few billion, and we are not the best on Earth in terms of length of our DNA. So, for example, red onion has 5x longer DNA than ours, and this Japanese flower has 15x longer DNA than ours. So it's really challenging. It's really heavy workload to be processed by computer, by current hardware, servers, cluster, and so on. And I'm pretty sure whenever you use some computer algorithm for any type of data for genomics or bioinformatics or metagenomics, you spend few hours, few weeks to wait for the analysis. And when we compare species, the same level, same kingdom, then the similarity is really high. So we are expecting around 0.1% only differences. And when we compare species from different type, like human and banana, for example, we have 50 to 60% similarity. And we could crack the first copy or the first sequence of our human in 2003, and later on we get an updated version of it, but that cost us a lot. So it took us 13 years to realize the first copy, and it cost us around $3 billion. So you can think about it, every character we can read it from our genome cost us $1. However, these days we are doing really good in terms of reading our DNA. And this is what we get actually, pieces of our DNA, not the entire content. And we are able to read 1000x more DNA characters in only 44 hours with less than $1,000. This is the state-of-the-art machines. And these are the sequencing machines that we are using. It could be hand-sized, could be attached to your iPhone, could be room size, fridge size, and so on. They are producing different workloads, such as read length, sequencing error, the cost, and so on. However, this read has no information about their order or the location of such segment in our reference gene. So we don't know whether this segment should be in the middle, in this chromosome or that chromosome. And that's why we need to link them together such that we build back our entire sequence. We got small pieces, we got long pieces, and we don't know which one of them is better because some of them really short but very accurate. Some of them really long, can be up to 2 million, but has a lot of sequencing error. So we are not confident about each character produced by these machines. So both of them are still used, short and long reads. And to do genome analysis, we got this genomic sample in chemical format. We send it to the sequencing machine. We got a bunch of reads. Then we would like to map each of these reads to a reference genome that we got in 2003 or any other version. And we got the first piece and try to stick it to the reference genome. We do the same for all these pieces until we get a very nice, smooth, full sequence of our DNA. And if it is a metagenomic sample, then we don't expect to have a single reference. So we have all of these reads. Then we move them to the database of references, not single reference. And then we try to match these segments to all of these species and try to find which one of these have high covered genome. So of course you should expect some shared read between all of these species. So the challenge here, how to resolve these ties, how to break the ties. If some of these reads map with high coverage to many of these species, then should you say something about all of these species or single one with high coverage and so on? Okay, what are the challenges in doing read mapping, for example? So whenever you have a read, and this is the reference genome, this is all the reads that you assign to certain location in this reference genome. So whenever you have a read and you map it to a certain location, the reference genome, you need to report all the location that this read mapped to. Why is that? So it might be all these locations that the same read mapped to having exactly the same number of differences, the same edit distance, the same cigar string. So which one to pick? You could apply some heuristics, but in some application, we really need to report all of them and later on decide based on the assembly or based on the applications such as metagenomics where we count these number of reads or overlapped reads. And we need to tolerate, of course, some of the differences between the read and each portion of the reference. Why is that? Because this is actually the main goal of this job, or genome analysis, is to know where are these differences and how many of these. So we need to tolerate them and have them here. And we need to do this process extremely fast because sometimes it can be life critical, especially for hospitalized children and so on. Okay, how we map read. So a brute force algorithm is just simply pick the read, pick the reference genome, and linearly scan the entire reference, location by location. But this is extremely expensive to do so. Why is that? So you have the read, and you have this location, the reference genome. You want to compare this read to here and tolerate some differences. How you tolerate some differences, you need to know to use dynamic programming algorithms such as Smith-Waterman, Needleman-Wunsch, and so on. And this will cost you m quadratic, the time complexity of such process. And how many reads do we have? Let be k, for example. So we have to bring all of these reads to map them to the reference. And how many locations do we check in the reference? N times, because we keep linearly scan. So that's why it's really expensive to do so. So we don't follow this method. What we follow basically in read mapping is the method of Yellow Pages book. So in my old days, I was using these books to find the phone number of my friend or colleague. These days, I'm not sure if it's there. We can search in Google, or you can just store it in your phone. So what we do in Yellow Pages, we go to the first few pages, where is the index there, and we check the family name, the first alphabet in the family name. If it is A, for example, we go to the index, what section has alphabet A? Then give you the page number. Then we go to the page X, where it has all the family names starting with this alphabet, and then we scan the second alphabet, the third alphabet, until we get the complete exact full name. And then we get the phone number. So this is simple 3 steps that we do for the Yellow Pages, exactly applied to reads, such that we map them to the reference genome. So the first step, we need that index for the Yellow Pages. How we build the index for the reference genome. So we have the reference genome, and we pick one segment from the reference genome, for example, the first 12 characters, and we call it seed or k-mer, and the k here is the length of this seed. And we scan the reference genome for this seed, and whenever it occurs in the reference, we record the location. And then we do this for the entire pieces extracted from the reference genome. And this is our index. So we use here, for example, this example, a hash table. So, but not necessarily to be a hash table. It could be any other data structure that can accommodate a huge amount of seeds with a huge amount of locations. And the good thing that we build this index only once for each reference. So whenever we have the same reference and we already built this index, then we store it somewhere and that's it. We don't need to rebuild it again. However, the size of this index really varies. So for human genomes, 3.2 gigabytes, this is the linear reference. But when you build the index, it really varies from 4.7 gigabytes all the way to 16.5 gigabytes. Depends on the algorithm that you are using. And this costs you indexing time. So it can go from 3 minutes all the way to 1 hour to build just the index for a good computer. And the differences in size really correlate with the seed that you pick. So it can be overlapping, non-overlapping, spaced, adjacent, non-adjacent, minimizers, or even compressed. So it depends on the way you pick these seeds, but at the end you need to really maintain high sensitivity, meaning that whenever I give you a read, you need to report all the location that that seed really exists in your reference. If you compromise the sensitivity to save some size here, then your read aligner is not really good. Okay, so after we build the index, exactly as what we do in Yellow Pages, then we have already the index and the reference genome. We pick one read coming from the sequencing machine, and we extract one seed exactly the same way as we do for the index. And we ask the index, I have this seed. Can you give me all the location that this seed occur in the reference in the index? So the index replies with this location that exists in the reference genome. So we go to the reference genome, and at each location, we extract the same length segment of the read, and we start comparing the read with that segment in the reference genome. And then we do some alignment or dynamic programming algorithm, same as we use in Yellow Pages, where we scan the full name character by character until we get the full piece of the name. And for some of them, they don't match, simply because they have more differences than a threshold. And this threshold is set by bioinformatician or by a user who need, for example, a threshold of 3. Then we define this similarity here exceeding the threshold, like having 4 dissimilarities, for example, or 4 differences. And if the exact match, then this has some meaning, so we report it to the user. And by dynamic programming, edit distance is minimum number of insertion, deletion, substitution, or differences that we have. And for having distance, we don't have insertion, deletion, so it's less challenging. But both of them are still used. So we have misfast, for example, read aligner, still using Hamming distance. And for some other aligner, we really need to know the insertion and deletions. So for example, organization operation, you can do simply calculating the edit distance and you'll get it 7 because we did 7 operation here. And this is the minimum number that we could do to make organization looks same as operation, for example. Organization and translation, you could do many other ways to make it exactly same as here, but we find that the edit distance is 4, not 5 as here or as here, because we're always looking for the minimum number of differences, which is here. Okay, and we published this first read aligner in 2009. This is very old, it's already get updated. However, regardless the read aligner we are using, even the fastest one, Minimap2 for example, here's the workload that is produced by sequencing machine. So one of the best sequencing machine that we have with high throughput can produce 378 million characters per minute. However, when we get the read from the machine and send them to our algorithm to do read mapping, the best algorithm that we have can only analyze 2 million characters per minute. So you can see the execution time bottleneck here. We have 150, at least 150x slowness between the read mapping algorithm and the read map and the sequencing machine. So we need to do something to accelerate this step, which is the initial step, by the way, to all the algorithm or all the application, almost most of these applications that we use in wet lab or for bioinformatics. Okay, and whenever a smart computer science student would like to accelerate something, he needs to know what makes it slow. So he needs to profile it and divide the execution time based on the component and know this is the component causing the bottleneck, so I'm going to target this very specific component. So we did exactly the same thing, and we observed 3 key observations. So the first one, when we analyze the execution time of the state-of-the-art read mapper, then we find around 60 to 90% of the execution time only spent on calculating the edit distance or the dynamic programming algorithm. And whenever we do dynamic programming algorithm or verification between 2 strings, we find that 98% of all these locations being verified are having very high dissimilarity or exceeding the threshold. So we're using dynamic programming algorithm, but those pair of strings that we already verify are useless because we're having high dissimilarity, so we simply discard them. So we are wasting our time using very expensive algorithm to verify those strings and having high dissimilarity. So we really need to do something to discover these highly dissimilar strings and remove them before being verified by dynamic programming algorithm. Okay, I kept telling you that dynamic programming algorithm is very expensive. We need to avoid it. We need to have a solution. But why did we use dynamic programming algorithm at first place? Can't we use just simply a linear time, for example, algorithm that can find these differences and that's it? So actually, dynamic programming by itself, it's really fundamentally beneficial for us. We really need it. We cannot avoid it. We cannot simply replace it by linear time algorithm. Why is that? Because when we compare Netherlands to Switzerland or any 2 strings, you really need to enumerate all possible prefixes when you do the comparison. For example, Netherlands to S, Netherlands to SW, and so on. So you can see a lot of repetition there. A lot of repeated computation is going on involved in the S, then the S is already involved in SW, SW is already involved in next, and so on. So to do that, we use dynamic programming algorithm, where we store the previously computed value somewhere in the cell, and when we need it later on, we don't recompute it again, but we just go and visit it and pick the value and use it again later on. For example, if I want to compute this cell of the table, I simply check this or that, and I do some computation here. That's it. I don't perform any complex operation. So that's why dynamic programming algorithm is very beneficial for me. But it has time complexity of quadratic time. So one thing about storing these previously computed results is that there are data dependencies. So I cannot accelerate this dynamic programming algorithm. Why is that? As I mentioned, for each cell, I need to revisit another 3 previously computed cells. So, for example, I cannot compute this row before computing that row, such that when I compute this, I need to have these 2 cells, for example, previously computed. So I need to build this row, then that row, then this row, and so on. Or I can do it column-wise, first column, second column, third column, and so on. I cannot do anything further to accelerate these things. Or I can do it anti-diagonal shape, for example. And the best way to parallelize such algorithm, either to have many cores or many threads, that each thread, for example, solving one problem of dynamic programming algorithm, such that I'm processing more than 2 strings in the same time. So these 2 strings processed by one thread of CPU or one core, and another 2 strings processed by another core of the CPU, such that I have parallelism between them. This is the best way I can do. And another observation, when you are doing global alignment, meaning that Needleman-Wunsch algorithm, for example, so you really need to compute the entire table and reach this cell, and that cell will tell you how many differences you have. So if it has a value of 5, then you have 5 differences, and this is the minimum number, between this string and that string. So you cannot stop in the middle, for example, and say, okay, I have 6 here, so I'm exceeding the threshold, so terminate the computation. I can't do that. I really need to compute the entire thing until I reach here. This is correct for global alignment, but if I use Smith-Waterman, for example, or semi-global, I might be able to stop somewhere in the middle. So it depends on the need or the design choice you have for read aligner. Okay, so we already profiled the execution time of the state-of-the-art read aligner, and we see what makes it really slow. And we figure out that all the problem coming from the dynamic programming algorithm. So hash table are really fast. Within O1, you can query the hash table and get a bunch of seeds very fast, extremely fast. You can still compress the hash table. You can improve the size of it. You can do further computation. You can combine FM index with the hash table as well. But the problem with dynamic programming tables. Okay, so what is our goal? Our goal is to reduce this execution time significantly using both new algorithms and a new hardware architecture. So if any one of you watched these 2 movies, Gattaca, for example, and Tomorrowland, the recent one, you will observe that they have a smartwatch, for example, where they put their finger and there's a microneedle in the smartwatch, take a little piece of blood, and then sequencing it, reading your DNA in just a few seconds or a small fraction of a second, such that it can identify you based on your DNA. So we would like really to have such of these devices in our life. However, to do that, we have open question. How and where to enable such fast genome analysis? And we don't need it to be just only fast, but we need to maintain accuracy, make it cheap. Cheap could be very expensive to us, but cheap by saving the life of the others, for example, or saving the inpatient cost. And we need it to privacy preserving, for example. I don't want to send my data or my family data to another company to analyze my DNA. So I want to maintain my privacy and do some exabyte scale analysis of genomic data. Especially if I'm in hospital, I'm targeting thousands of patients in the same day. So I really need to do it at scale, not one by one. Okay, so this is today a conventional computer that we have, even laptops, mobile phone, and so on. What do we have is a storage place where we store our data and a fast memory that can move some data here and buffer it to the CPU. So I have a CPU where I process all of my data. I cannot run an algorithm here or here. I should run it on the CPU. So I should move data from here to there. And this is the sequencing machine that produced the read. And what we do as a bioinformaticians, so we move the FASTQ file coming from the sequencing machine to the hard drive or the SSD, and then ask the CPU algorithm to execute some analysis on my data. So the data will be moved from hard drive to main memory to the CPU processor. And this is very time consuming, very energy inefficient, because these buses between these components are very power hungry because they have very high throughput to move huge amount of data from here to here in very short time. So what we would like to do is to change this architecture. We don't like the current architecture of these CPUs. These CPUs as general purpose computing, they are doing really good for some applications. But this is a huge workload and many other huge workloads such as machine learning, artificial network, deep learning, and so on, where you have huge graphs and you want to train them and process the data. You need something very specialized to do that. So by specialization, we mean we don't want to move to SSD, for example, first. We could move to the memory if we can do some computation here. And that's why we call it hybrid main memory or 3D stacked memory. Or we want to process the data to the FPGA or to the GPU. So all of your laptops or your mobile phone have their own GPU to process some videos, some pictures, some image processing, and so on. So if we could process these data directly to the GPU or the FPGA or the CPU, and these are different architecture, different computing architecture. You don't need to know the architecture of these, but just think about it as different type of CPUs, for example. But they are extremely fast. And we could also process all of these things inside the sequencing machine. And this is our goal in our research group, is to do the processing inside the sequencing machine. We don't want to wait 1 day or 2 days until we get the data from the MinION or whatever sequencer you are using. Then after 2 days, you send the data, which is a huge FASTQ file, to your CPU, and then wait another couple of days to analyze your data, which you already wasted 1 week to process it in wet lab and isolate the DNA molecule and so on. So we don't want to do that. We want just to get the sequencer ready, and within a few minutes, it will process a chunk of your data, it will output a piece of your FASTQ file. Then we want to process it on real time, on the fly. We don't want to wait to get the full package, the full file. So processing chunk by chunk of this data, and whenever it finishes, we already processed most of the data we have in hand. So within just 44 hours, you can finish sequencing and read mapping, for example. And we know that most speed up comes from parallelism. So sometimes the algorithm is not by itself, such as dynamic programming. We cannot parallelize it. We cannot do any computation in parallel by itself. So what we can do parallelism enabled by novel architecture and the algorithm. You can build a huge chip where you can process many cores inside the same chip, and each one of them doing a specific dynamic programming algorithm, for example. This way you can accelerate it. And this is true even for this Japanese preferred network company, where they have this specialized core called MN core last year, where they have 4 dies, 4 pieces of the processor, where each small piece of the processor can process simple arithmetic computation. And they use this to accelerate deep learning, for example. So they already realized the importance of doing some specialized computer to accelerate certain problem that we really need to solve. Same thing for another company who produced this huge chip for accelerating machine learning application. So this is the very latest GPU we have, and this is the chip size. So you can imagine how large this chip compared to the GPU. So they said it's okay to have very large chip because our workload, machine learning application, is really huge, really varies from different applications. So this matches our needs, for example. So it's okay to have a larger one. It's okay to have more expensive one than the CPU if we are able to process it in a very fast way. This is another French company called UPMM, where they have this DRAM chip that you plug it in your motherboard or CPU or laptop. But what they do, this is very regular, normal DRAM. You can have 4 GB or 16 GB to save some data between the SSD and the processor, for example, or buffer it. But what they did, they add some processing cores inside the DRAM. So you don't need to wait to move your data from the DRAM to the CPU, for example. You can do simple bitwise operation inside the DRAM. And these are already available. So you can leverage them, for example, to do some computation. And this is another company called Tesla, where they produce this autonomous driving car, and they produce this customized hardware. So this piece of hardware having two chips, and these chips doing exactly the same job. So they just have redundant piece of hardware. Why is that? For safety and reliability. If one of them failed, then the other one can operate safely, and you don't have any problem with driving. So all of these companies realize the need for having specialized hardware for the problem at hand. And it's okay, we pay 5x higher to buy this car, but we really need it, and we feel the fanciness of having these cars. So exactly the same thing can be applied to genome analysis. Buying extra little piece of money for a sequencer that can do processing inside the sequencing machine, for example. This is another news happening in the last year, where Illumina, which is a company producing those short trees, or the sequencing machine that produce these short trees, they acquired Genome. Genome is a company that having FPGA chip to process some of the bioinformatics application. So you can think why they acquired such a company, why they need FPGA, for example. So maybe in the near future, in the next 10 or 20 years, they might have FPGA cards to do some processing, some read mapping inside the sequencing machine. Yeah, most of these today sequencing machines, they already have FPGA, but it's for maybe for base calling or to do some processing related to sequencing, but not read mapping, for example. So when they have such a company or they have such power, you can think about we will have maybe in the near future, this sequencing machine can do all type of bioinformatics pipeline for us. This is another news happening the same year, and where Illumina is negotiating with Backfile, which is another company producing sequencing machine with very long length and high sequencing error. So this deal is not set yet, so they're still negotiating. Maybe they cancel it, I don't know. But you can think if Illumina get the power of FPGAs and the power of Backfile, then they might have some sequencing machine that produce short and long, or both of them at the same time, while they can process them inside the machines. So it's very promising to do such thing. Okay, so now I show you the problem, the real problem we have, and I show you some insights from industry, how they can accelerate other problems. Okay, what other researchers are doing to accelerate such a problem? So we have 3 key directions to accelerate read mapping. The first one, seed filtering techniques. Second one, pre-alignment filtering. And the third one, accelerating the dynamic programming algorithm. And it's okay, I'm going very fast through this slide, but I'm going to explain it in the next slide with example. So back to our old example, where I have already the index and the reference genome, and I got a piece of read. So I want to get a seed out of this read, send it to the hash table, get a bunch of locations, then go back to the reference genome to extract these segments at these locations. But what I do, I don't go and check every single location in the reference genome. I did some seed filtering heuristic to filter most of these locations. And only some of them will go to the reference genome and extract few segments from the reference genome and then compare them together. But I said, okay, stop. Before doing dynamic programming algorithm, I'm going to apply some filtering heuristics to compare these 2 segments together. And if it really matches, then I apply dynamic programming algorithm. But if it is not, if it has a lot of dissimilarities between the 2 strings, then I don't apply dynamic programming algorithm. So by doing seed filtering, pre-alignment filtering, and acceleration for dynamic programming algorithm, I cover all type of acceleration you could do to the read mapping. Okay, so now let's cover the seed filtering. So this is a work we published in 2013, where it's called fast hash. So it has two main observations. The first one, we call it adjacent k-mers. So by adjacent, meaning that if you have this piece of read and you have this reference genome, and this read, you can get 3 adjacent seeds. So let this be a seed, this is another seed, and this is a third seed. All A's, all C's, all T's, for example. So I pick them by nature adjacent in the read. So I expect them to be adjacent as well in the reference genome, because it makes more sense for me. So when I send these seeds to the index and I ask the index to provide all the location that each seed exists, so the hash table give me this location, that location, and so on. So the problem, for example, this seed, which is the red, the first one, and the yellow one, which is the second seed, they are really apart from each other. So there is a huge distance between them. So it doesn't make sense that my read mapped to these very far seeds. So I choose the location that are adjacent to each other and cancel all the other locations, for example. And that's why we call it adjacency filtering. Now, these don't have to be necessarily like very adjacent to each other with 0 space. They could have some certain space, because at the end I need to allow for some insertions and deletions. So if I have insertions, I don't expect them to be next to each other, they should be apart. If I have deletion, maybe I don't expect to have the 3 seeds next to each other. Maybe I have 2 of them and the third does not exist. So all of those are parameters, you can play with them, but the idea is still there. The second observation, we call it cheap k-mer. So by having this reference genome, and then I check pieces of it with the index, I got a bunch of location for each seed. So this seed give me a location list, or list of locations with 4 locations, here 2 locations, 1,000 location, 2 locations, and each list coming from different seeds. So this list is provided by the hash table or the index. And what we observe that most of the seeds that have many occurrences or many frequency, high frequency in the reference genome have no meaning. They provide no specific or no useful information as a seed alone, individual seed. But the read itself might have some specific or scientific information. So what we said, okay, let's have a threshold equal 500, for example, and whenever the list of location exceed that threshold, then we just ignore that list because it's really expensive to verify. However, this is still heuristic, and the final say to say if this is a good heuristic or not is to preserve very high sensitivity. So we will not, when I give you algorithm, a read, I want to get all the correct location in the reference genome. So you could apply different heuristics. For example, I don't want to ignore the entire list. I said, okay, I will apply the threshold to only to the first 500 location, and the rest of them I will just ignore. Or you can combine two lists, for example, and you can sort them, all of them, and apply adjacency filtering first, then the cheap k-mers. But adjacency filtering requires you to know that these seeds exist after each other or not. That's why you need sorting. And sorting by nature, it's really expensive to perform. So all of these are design choices, just to reduce the workload for the dynamic programming algorithm. And here, like the numbers, you can reduce this much of location to only these locations that can be verified by dynamic programming algorithm. And this is the paper, we call it fast hash. So I'm going to skip the conclusion. Here's the link where you can download the source code, you can play with it, you can have the entire algorithm, you can set different thresholds and test it, and see if you can get less sensitivity, or you can compromise it, or you can maintain it accurately. Okay, so let's move to the second direction, which we call pre-alignment filtering, which is applied just right before the dynamic programming algorithm. And please note the difference between seed filtering and pre-alignment filtering. Seed filtering, we are playing with the output of the hash table, just list of location. We are trying to shorten these lists. But in pre-alignment filtering, the input is really two segments of text, not the seed location. So we got those two strings, and we would like to verify them using dynamic programming algorithm. So again, we notice dynamic programming algorithm is expensive, and our goal is to reduce the need for dynamic programming algorithm. We cannot avoid it, we cannot replace it, but we can just reduce the workload. So we propose different type of pre-alignment filters that each of them can filter different strings. If we notice that really they have high dissimilarities, then we delete them. If they are equal or if they are similar, then we send them to the dynamic programming algorithm. However, we need to observe these three important things. We need to keep them. So the first thing, we want to delete most of these highly dissimilar strings. We can't delete all of them because we are using heuristics. If we are using very accurate algorithm, then it will be very expensive as dynamic programming, for example. And we want to preserve all the correct one. Whenever we have two pieces of strings, they are similar, we want to keep them. And similarity here doesn't necessarily to be exact match. It should be within a threshold. And we want to do that quickly, of course, because our goal is to accelerate this step. So if we are spending too much time in filtering, then we are increasing the entire end-to-end execution time, for example. So this is the first work we proposed in 2017 called Gatekeeper. And what we do in Gatekeeper, we observe that if two strings differ by E edits, by E differences, then we could align them. We could know very fast how many differences they have using just 2E shift. How we do that, I will explain in the next slide. But the key idea here is we compute some several bit vectors, and we use AND operation with all of these vectors to tell you very fast way how many differences they have without computing dynamic programming algorithm. And this is the key results we get from implementing it in FPGA. And I'm going to tell you why we choose FPGA as a processor. So it achieved 90x to 130x faster than SHD, which is another pre-alignment filter, and the adjacency filter, which I presented just in the previous few slides. And when we add Gatekeeper, for example, to existing read mapper, Mr. Fast, we achieve 10x end-to-end speed up. Okay, what is Gatekeeper? What is the algorithm behind it? So let's consider these two words, Istanbul and Istanbul, and I would like to compare them. Now let's use Hamming distance. No insertions, no deletions. If I do just XOR operation or just comparison, pairwise comparison, character to character, then I get exact match, 8 matches. Each character matches the other one. But now let's do ourselves single deletion or single operation. Now let's delete this character, A. And now when I delete the character, all these trailing characters will be shifted to the left, such that the word gets changed to this, right? So all of them shifted to the left. Okay, now if we do another Hamming distance or comparison, we will find that there are 5 mismatches, not single one, although we just deleted one. So how to fix that? What we said, okay, if those characters are shifted to the left, now let's shift them to the right by ourselves such that we can fix this problem. So we make another copy that is the same copy, the one we modified, but shifted to the right. And now we do the comparison with these two copies together. So when we compare XOR pairwise comparison, then we get 4 matches and 3 mismatches from there. And then we combine the results. So this is XOR, providing you a bitwise result. And this is another XOR operation, so we got those 2 bit vectors. And now when we perform AND operation, why we choose AND? Because we are looking for these zeros. So 0 always dominate and cancel the 1. 1 times 0 is 0. This is how we get it. So at the end, we receive that there is a single difference here at this location, which is actually correct. So we deleted the A at this location. And when we count them, we got 1 mismatch rather than 5 mismatches. Okay, this is a very toy example, simple one. And now when we move to real example, we don't really know where are the deletion, where are the insertions. So because in the example, we know that we made the deletion, so that's why we shift the copy to the right. But in real life, we don't know how many insertions, how many deletions, how many substitutions we have. So we cannot shift to the right or to the left. We need to consider all combination of data. So we have this here a query and here a reference, and we want to compare them together. And what we generate, all of these bit vectors, how we generate them. First of all, how many of them we need? We need 2e plus 1 many. And what is e? E is your edit distance threshold, the same that we are using for dynamic programming algorithm. So in this example, I choose it to be 3. So I have 7 of these bit vectors. And how I generate them, so exactly the same as the toy example. The first one is just pure XOR operation between the two strings, just XOR. If this match, then 0. If it doesn't match, then 1, and so on. Now the second one, or these 3 bit vectors, the second, third, and fourth, how we generate them, we call them first deletion mask, meaning that this one, we shift this by 1 step to the right, then we compare it to this using XOR, and we get this. And for this, we shift this by 2 steps to the right, then we compare it to this, and we get this. And so on. We keep doing this until we reach the threshold, which is a 3. So we do 3 bit vectors for deletions. And for insertions, we do exactly the same thing as deletions, but in the opposite direction. So we shift this to the left, and then we compare it with this. So we got this bit vector. And here, two steps shifting, 3 steps shifting, and we got this one. Now, why we do shifting in both directions, left and right? This is just to cover whatever cases you can have. You can have single deletion, then 3 insertions, then 4 insertions, then 5 deletion, and so on. So as long as it is within your edit distance threshold, those should cover all potential cases that you might have. Now, in this very specific example for these 2 texts, this is the correct result. So these are the correct matches between the 2 texts. If you notice here, it's really small, but you may focus here. You can see this portion exactly similar to this. That's why we got it in the XOR operation. And here another difference between the T and the G. And then after that, followed by a large number of matches. Then here, we perform a single deletion. For here, there's single C, but here 2 Cs. So we delete one of these Cs. That's why all these zeros appear in the second mask, not here, in the second bit vector. So this is the correct answer, actually. All of these number of consecutive zeros. So by counting their number, we got how many similarity or how many matches between the 2 texts. And by counting the ones between them, we get the number of differences. So this is 1, 2, 3, and 4 differences in here. And that will tell you if these 2 texts exceeding the threshold or less than the threshold in extremely very fast manner, much faster than dynamic programming algorithm. And then we do AND operation to track all of them. But the problem with the AND operation is that always the 0 will dominate whatever you have vertically here. So whenever we want to count these, when we do AND operation, there is a 0 here. So it might cancel this one when we do AND operation vertically. So to fix that, we did some heuristics such that convert all single 0 or 2 zeros into 1s, such that when we do AND operation, we no longer have all these random zeros. And when we do this heuristics, this is what we got. So you can see each bit vector has less number of zeros. We cancel all the single 0 or 2 zeros. And then we do AND operation. And we got this result of the AND operation. You can see we got 3 mismatches rather than 4 mismatches. So this is okay. It's okay to have false positive, but it's not okay to have false negative. It's not okay to overestimate the number of ones, for example. And by doing AND and reducing always the number of ones, we are guaranteed to have 0 false negative. We are always underestimating, but not overestimating the number of ones. And this is the correct result we got in from Needleman-Wunsch, for example, dynamic programming algorithm. It shows the mismatches at the exact location as Gatekeeper shows, for example. But we really don't care about the cigar string or the location of the difference from Gatekeeper point of view. Gatekeeper only addresses or answers the question, will your 2 strings exceeding your threshold or not? That's it. And then if it is not exceeding, you send it to dynamic programming algorithm, which will provide you alignment score, cigar string, and all those needed information. Okay, so you could ask me, what's the difference between your algorithm and dynamic programming algorithm, for example? So in our algorithm, we do just pairwise comparison, regardless the shift process. So we just apply XOR between the two individual characters. So this one, for example, we did the XOR operation to achieve this. So just pairwise comparison. But for here, we are shifting this one such that we can compare it with the second character, not the first. So we are shifting this by one step, then we compare it with this so that we can get this diagonal. And we do this same thing, but we shift this by two steps such that we compare this not with the first, but the third, and so on. So by doing this, there is no data dependencies between those cells. So each cell here is computed really independent of any other cells in the diagonal. But here, we really need at each cell 3 pre-computed cells, so that we need one row followed by another row and so on. But here, everything can be computed in the same moment. So our goal after we build this matrix is to track these consecutive zeros. So we want to count how many consecutive zeros here and there, so that will tell you how many differences you have. And to do that, this is bitwise operation. So for those who might know about FPGA or GPUs, it's really very cheap to implement such an algorithm, because just XOR, shift, and AND operation. That's it. So you can achieve all of this in a single clock cycle. Not like CPU, which you need sequential computation. So you can choose the computation platform you like to implement such algorithm. And we choose FPGA. Why is that? Because FPGA, it's like a grid of lookup tables. So this is FPGA chip, where you have a large number of pieces, and each piece called lookup table. So you have many of these pieces in FPGA chip. And each piece of this, called lookup table, can process any kind of logic computation you need. If it can fit in single lookup table or many of them. So it can implement for you the function you like. And for that reason, we choose FPGA, because it also depends on the bitwise operation. So this is how we implement it. So every single lookup table, we make it responsible for 5 bits, such that will produce single bit in the output result. So here, within 5 bits, we're computing it at a very fast speed, and we have many of these lookup tables, such that each one will process overlapping 5 bits and produce one here, which is not overlapped with any of these bits, such that we process all these bit vectors in the same time, because all of these lookup table working in the same time, not like CPU cores, for example. This is why I choose 5 bits, not 4 bits, but at the end it's design choice, and depends on the FPGA you have in hand. So some of the FPGAs having a lookup table with 5 input, or 4 input, or 3 input. So that shows you the importance of knowing the hardware before you design something. So just implementing an algorithm or an existing algorithm and send it to the FPGA doesn't guarantee to give you high speed up, unless you really know the hardware you have in hand. And this is the FPGA we have. So all of this is the FPGA, where we place many cores, as I mentioned for the dynamic programming algorithm, and each core can achieve one filtering independent of other filtering, such that you can verify many strings, many pair of strings in parallel. And then whatever accepted by the filter, you can send it to the dynamic programming algorithm. So in this method, you are not compromising anything. You're still achieving everything you need from Needleman Wunsch, for example, and you are achieving very fast filtering using Gatekeeper. And this is the utilization, how many of these pieces we are using as part of the FPGA. You can notice here, we can use all of these available lookup table as well, but when we move the data from the sequencing machine to the CPU, then from the CPU to the FPGA, because this FPGA need to be plugged in the motherboard. So we have no other way to transfer the data without the PCI Express, for example. So the PCI Express only provide data for this lookup table. So if we have our design inside the sequencing machine, we could fit all the data to the FPGA board or the FPGA chip such that we can fully utilize this FPGA chip, for example. So our design is extremely cheap to be implemented on FPGA because the operations are very simple, and all of these can be implemented on FPGA without high utilization. And I'm going to show you next, in the next few slides, the utilization of the FPGA. But anyway, the source code is published here. You can play with the CPU implementation, with the SIMD implementation, with the FPGA implementation. And these are the results as I presented in the conclusion. And let's skip this. And this is the paper. And this is another implementation of the algorithm, but in SIMD instructions. This is the paper. And now the question is, can we do better? Can we have a scalable design? So in this design, as you can see, all these bit vectors need to be ANDed together as one piece. So we bring the full bit vector, then we AND it with the next full bit vector, and so on. But this is not scalable. So if I change the read length from 300 to 10 million, what will happen to this AND operation? Do I really need the entire 10 million bit vector to be ANDed with the next, and so on? So this is the paper we presented to address this issue. We call it shoji. So shoji is a Japanese term, which is this, meaning the sliding door. So what we observe here, as you still remember the green segments in the bit vectors, the green segments of consecutive zeros, where we want to count them, how many of these zeros we have to know the number of similarity and dissimilarity. We notice that they are always long, and they are next to each other. So we said, okay, why, rather than ANDing them together, why not just go to the bit vector and start counting how many zeros we have in individual bit vector? But this is challenging because there are correct zeros that are part of the solution, and there are many other random zeros in the bit vector. So what we did is we have exactly the same thing as we have in Gatekeeper, where we generate this middle diagonal using just XOR, and this is by shifting one of the tags and then compare it, and this is shifting 2 steps and then compare it. And what we do is we said, okay, let's have a search window of length 4 bits or wide 4 bits. And now we are counting in diagonal shape. So we start counting the first diagonal. We got 1 bit, 1 bit, 1 bit of 0, 4 bits of 0, and so on. So we're just counting number of zeros in each diagonal. And then whenever we have a large number of zeros, we said this is a greedy choice, that this is probably part of the solution because it's long. So we always observe that these zeros are long, much longer than the random zeros we have overlapping with the correct solution. So we pick the 4 bits and we store them somewhere. And then we proceed to the next search window by shifting it just single step to the right. And then we repeat exact same computation. So we said here's a single zero, single zero, single zero, and here are there 3 zeros in this diagonal, no zeros, 2 zeros, no zeros. So which one is the longest? We pick the one with 3 zeros. And now we want to store it here. But remember, we are sliding this window by single step, and the width of this window is 4 bits. So there are, every time we count, there are 3 bits overlapped with the previous window. So when we want to store this result here, there are these 3 bits that overlap between this window and the next window. So which one to store? The old one or the new version? So we always choose one of these 2 based on the number of zeros. Which one will increase the number of zeros stored in this bit vector? So if you notice here, there are 3 zeros and there are 3 zeros here. So here it doesn't matter. You can store any one of them because the number of zeros will remain the same. So we store it here. But if this is 1, for example, here, then we don't store the 3 zeros or the 3 bits coming from this. We store the older one. Why is that? In this way, we are always underestimating the number of ones because we are increasing number of zeros. This way, we guarantee no false negative at all. And as I mentioned earlier in my slides, that the accuracy really matters here. We really want to maintain high accurate algorithms without affecting the accuracy of dynamic programming the algorithm or read mapping step. We keep doing this for all the search windows we have here. And remember, each of these search windows independent, totally independent of any search window in this matrix. So you can process these 4 bits or this search window independent of the first one, as well as all of these search windows. So all of them can operate in parallel using just 4 bits of piece of information coming from the bit vector. So this is the main difference between this work and Gatekeeper. In Gatekeeper, you require the entire thing to be there. But here, you just require 4 bits of them to be within a search window. And all the other bits, you don't care about them because they will be processed by another search window. And then we simply here count the number of ones. We get the number of differences in a very fast way, much faster than dynamic programming algorithm. Okay, any question here? Is the algorithm clear? Questions? Okay. So that's why we call it shoji, because we use this fashion where we slide the search window over the bits. And this is why we choose a window of size 4, not 3 bits, for example, because it has a reason that what's the minimum number of matches we could have? What's the minimum number of zeros we could have? So the minimum number is always single one, right? This is the minimal you could have. And to be minimal is you should have 1 around it, right? Otherwise, it's not minimal. If it's 3 bits of zeros, then it's not minimal. So to have this pattern, we can have a window of search window of size 3 bits, for example, right? To cover this pattern. But when we notice here, we find a lot of 1, 0, 1 that are not part of the correct solution highlighted in yellow. So you have a lot of random zeros. For example, here 1, 0, 1, here 1, 0, 1, and so on. You have a lot of them everywhere. So to avoid these to be counted in the correct solution and increasing your false positive, we said, let's increase this search window by a single step such that we include one of these bits, either here or here, based on the solution. So we add 1 here or 1 here, it doesn't matter. At the end, you will search for 4 bits only. And when we try this practically, it really decreases our false positive. And we implement this in FPGA. Again, FPGA, you can think about it as just processor, a normal processor you have in your computer. But the good thing about it, that is highly parallel, and you can reconfigurable. You configurable all things with inside the processor, for example. You can tell the architecture that I want to use this piece or this piece rather than this piece, for example. But in CPU, you cannot control it. You just send the code and let the processor processing it. That's why it's more general purpose. But for this one, you can specialize it by tell this portion of the FPGA do this operation, here do another operation, and so on. So implemented in FPGA such that we have search window here, search window there, another one here, another one here. So all of them work in parallel, and you can process the entire string in a very fast way. Okay, let's skip this. And this is the paper. This is the link where you can get the CPU implementation, just code implemented in C, and as well as the FPGA design that we use. So you can play with it, you can change the mechanism, you can change the parameters or develop your own ideas. And the last one for the pre-alignment filtering, we call it Sneaky Snake. This is a recent one, but we implemented for all available hardware that you can imagine. CPU, GPU, FPGA, all of them. We try to leverage which one will provide you the most speed up out of Sneaky Snake. And the key observation of this work is that we notice these green segments are always non-overlapping. So by reading them sequentially, one by one, we can observe a correct counting of number of consecutive zeros, rather than going to the overlapped window, for example, as Shoji, and count how many overlapping zeros we have. No, here we said, okay, we want to scan all the number of zeros from left to right or from right to left. And for that, we said calculating edit distance is really similar to finding the problem of single net routing problem, which you can think about it as shortest path in this maze. So this is what we call VLSI chip, which is actually any chip you have it in any electronics, which is the black piece, you have the square piece in any electronics. It has legs to the right and legs to the left, something like that. So this piece of electronics has a number of inputs here, a number of outputs. And if you want to connect this input such that you send your input data to somewhere here as a processor, or you want to assign it to another output here, for example, so you cannot send it directly here to there, because there's an obstacle here. So you really want to avoid this obstacle, and you go from here. So this is what they are looking for in the industry, for example, to find the cheapest way to connect this link to that link, because this will cost them money to have longer wire or shorter wire, because they do this as scale. So we leverage exactly the same problem where we have the VLSI chip, for example, here, and we want to go from here to here through this maze by avoiding a number of obstacles and make it short. So now let's explain the example, and this is the result actually what we got. So this has an improvement up to 4 orders of magnitude less false positive compared to Shoji and Gatekeeper. It's really a huge improvement in terms of accuracy. And by doing this just for normal computer, there's no cluster, no hardware acceleration, we got 37x speed up compared to idlib. Idlib is the best implementation for Needleman-Wunsch algorithm, for dynamic programming algorithm, as well as Parallel, which is very slow, much slower than idlib. And when we implement this in GPU and FPGA, we get this much of speed up compared to idlib and Parallel, which is dynamic programming algorithm. So imagine you having 600 or 400x of speed up compared to the dynamic programming algorithm you are having. Okay, so back to our matrix. And this is exactly how we compute it as Gatekeeper and Shoji. And what we do now just for simplifying the example, let's lay it horizontally rather than diagonal shape. So you're converting exactly the same values into this. So this is the main diagonal, this is the one shifted by 1 step, 2 steps, 3 steps, and so on. So we convert this into this. Now imagine with me, each one is an obstacle. Each one is black box, and the zeros are white boxes or available path where you can detour your signal. So from this, we got this. Okay? So we didn't change anything. This is just a way of representing the matrix. Now, this is your entrance, and this is your exit. And you would like to go from the entrance to the exit with the cheapest cost, or the cheapest path, or the shortest path. Depends on the way you define it. And how we define a cheap path? By having less number of obstacles through a certain path. So how we solve it? Remember, we have the edit distance threshold as 3. That's why we have 7 rows. Remember, this is our number of bit factors. So 2e plus 1. That's why we have 7 rows. Now, this is our snake. This is our friend. And the snake would like to travel from the entrance to the exit. So what we do? The entrance will go to each row and try to check if this row has available path or not. So the first one may have 0 available path, or 0 steps, 0 steps, 0 steps, and here I can move 4 steps. And the next one, I have single step, 0 step, single step. So which one to go through? Which one I can travel through which path? So as a greedy choice, I always need to choose the one which has had the longest number of steps, or the largest number of steps, or the longest path. By having always the longest path, I guarantee that this is the shortest one, because it's always having the least number of obstacles. And we have the proof for that in the paper. So we go through number 4, or the one having 4 steps, and we avoid the obstacle at the end. Why we avoid it? Such that we can jump on this obstacle and continue iterating over the algorithm. So now we reduce the 3 into 2 because we already avoided this obstacle. So I'm allowed to avoid 3 main obstacles, basically. So I jump over the obstacle and now iterate again. Exactly the same method. So here, do we have an available step? No, no. I have 4 steps, no steps, 2 steps, and 0 steps, and so on. Now, which one to choose? Of course, I would choose the longest. So I will go through the 4 steps until I reach an obstacle. And when I reach an obstacle, I reduce the number of available obstacles to 1. And now I repeat exact same computation, but here I will find the ties. I will find the path with single step, and single step. Which one to choose? It doesn't really matter for me. So all of them having the same value, same length, same number of obstacles. So which one to pick, it's up to you. So I'm going to pick any one of them. And then I have an obstacle here which I avoided. So I'm going to reduce the number to 0. Now, what does this number really mean? So this number is basically the number of differences between the 2 texts I used to build this matrix, right? So if I reach the exit with 3 or less avoided obstacles, meaning that these 2 texts or these 2 strings having less number of differences than the threshold, then this is okay for me, and I'm going to send those 2 texts to the dynamic programming algorithm. You got it? Okay. So any questions so far? Should I repeat it again? Should I? Okay, very simple, right? You just count the number of available paths and you jump over the obstacle. However, this is not the only optimization provided by SneakySnake. What you really need to build is not the entire matrix. What you really need to do is this one. Not this, is this. What's the difference? So you really don't need to compute all these cells, these, and these. Why is that? Because whenever I start counting from this side, so I see an obstacle here, then I stop, because I'm not allowed to go through an obstacle unless there are available paths here. So when I observe an obstacle here, so I stop computing, I go to the next row, stop computing, next row, stop computing. But when I see available path, I keep computing until I reach an obstacle. Then I stop, then I move to the next, compute, obstacle, stop, obstacle, don't compute, available path, move 1 step, you find an obstacle, stop. And then you iterate again. So you save a lot of computation here, and that makes the algorithm extremely simple. So the number of computation here is significantly uncomparable to the dynamic programming algorithm we are doing. So that's what makes the speed up really huge compared to idlib, for example. Any questions?
How do you compute this using SIMD? Like, where's the parallelism?
Okay, so for SIMD is a bit challenging.
Okay.
So in SIMD, what is SIMD actually, for those who don't know? SIMD instruction is something where you do single instruction, multiple data. This is what it stands for. And what this does mean, so you have a line where you can think about it, you have this lookup table exactly here. For example, 4 lookup tables. And this line will be executed regardless the amount of data you have. So the best way to fully utilize this line is to have all of these lookup tables working at the same time, for example. So to achieve this, for example, using SIMD, what you really need to compute is the entire thing, because you don't know where are the zeros, where are the ones in advance. So you can't save the computation as we did here. So you can implement the entire line here and then count how many zeros, and then the entire next line, third line, and so on. And then you count. But the best thing in SIMD, you are doing all of this in parallel. So you might achieve the same speed up as we achieved here. This one we implemented using pure C. There is no vectorization, there is no SIMD instruction, there is nothing. Just a pure sequential algorithm. And when we compare it to idlib, which is parallelizable, this is extremely fast. And the other thing, we really don't need to store the values here in any register or any data structure. All of this computed on the fly. You don't need to store it even, the entire matrix. Because you just here to compute this, you check the first character of the first string and the first character of the second string. And if they don't match, then they just ignore and move here. Then you get, so let's explain it here using a different color. This. So this is the two strings. Can everyone see? This is the other string. So to compute the first cell, we compare this with this. And now to compute this, we compare this. We compare the first one with the second one here. And then the first one with the third, and so on until we get an obstacle, for example. But to compute this, you can compute the first one with the second one here. And by doing this, we don't really need to store anything. What we really need to store is this, only this value. And we keep reducing it every time we face an obstacle or a mismatch in the long or the shortest path. And this will tell you the number of differences you have. Any questions so far? Okay, this is the number of lookup table that we are using from the FPGA implementation of the 3 algorithms. So you can see here the number of algorithm in percentage in terms of the total number of available lookup table is really less than 1% almost. So less than 1%, meaning that you can replicate, you can have a duplication of many cores in the same chip, as many as this can be fit into here. So if it is less than 1%, then you can fit probably around 100 of these cores, such that each core can process 2 strings independently from other cores. And if you are using a larger FPGA, more expensive one, you can fit more of these cores such that you can meet the needs of the hospital or the university and so on. This is the paper, again, the source code available online for the 3 design, GPU, CPU, and FPGA, so you can play with it as well. Okay, so we design all of these pre-alignment filtering, and what we notice in all of these kind of work, that there is a problem bottlenecked by data movement. So we are always moving the data from the sequencing machine to your CPU, and CPU will be moved from the SSD or the hard drive to the main memory, from main memory probably to the processing core inside your laptop, or to external FPGA or to the GPU. But still the data movement is there. And that is expensive. So we really need to design something that can do processing without moving the data back and forth. And this is a paper called Google Workloads for Consumer Devices or Mobile Devices. And this paper, they analyze for Google browser or for Chrome, what is the energy efficiency of such workload. And this is the main memory you have inside your computer. This is the processor. This is the processor chip, but it has also caches, what we call a cache, which is, you can think about it as very fast memory, but with very small size. So we have many caches here, and this is the CPU core, which can be FPGA or GPU. This is what you have inside your laptop. And then we are moving data from the main memory, and before it, we move data from the hard drive to the main memory, to the caches, to the CPU. And they observe this data movement cost the total energy by 62.7%. So you're spending more than 2/3 of the total energy budget just on data movement. There is no computation here. Just moving the data from the DRAM to the caches to the CPU and then back and forth. This is really a huge amount of energy and time you are spending just by moving the data. So rather than just focusing on accelerating your algorithm for the CPU, you really need to be careful about data movement. So it's always the best algorithm where you can fit your data inside the memory or inside the cache. This is very optimal. But if you keep reading the file from the hard drive, storing the result to the hard drive, go and read back, this is wasting a lot of time. Even if your computation is very simple, but you'll be spending most of the time on just I/O, reading input and output from the hard drive. So we try to avoid this. And one potential solution is to move computation close to data. So if the data is here, provide some processing cores inside the memory, for example, as the French company did, which called UPMem. Or you can provide more memory capacity to the processing cores. So all of these directions having its own challenges, its own difficulties to do. But if you can achieve this, even for the conventional computer that you have, it will be great to have it. You'll save a lot of energy, a lot of time, even without optimizing computation. Of course, we have limited area and energy budget. So those devices know how to cool themselves without all of this overhead. But when you add computation to the DRAM, you don't expect to have exactly the same design for these laptops because you need more cooling power for these DRAM because they will overheat very fast. And this is one of the solutions proposed by one company called Micron, where they have HMC cube. HMC cube, so this is normal DRAM. This thing is the normal main memory you have in your computer. And what they did, they said, okay, the main memory, we cannot advance it more because the scaling technology node is not going further. What we can do is we have this DRAM chip, let's have another one of it on top of it, another one, and we can stack many of them in 3D, such that we can have this cube. So each layer of this cube is a DRAM. And all of this is integrated in single chip, so it's single piece, such that we avoid data movement between different DRAMs. So if all of them the same die, same chip, then we don't need to move the data from one piece to another. And they are connected together by very high speed links, such that they can achieve more than terabits per second. And the more interesting thing, the very last layer, so all of these are DRAMs, but with the very last layer called logic layer. And the logic layer is exactly as this, which has a huge number of lookup tables. So you could do the bitwise operation or more complex one, the one you like, inside the logic layer, where you have a huge amount of memory bandwidth on top of it without the need for a processor. Or you may be needed for very complex operation, but most of the computation will be done inside the memory without the movement to the processor. So by utilizing such technology, you can achieve very high, very advanced algorithm for read mapping, for example. And this is what we did next. So in this filter called Grim filter, we applied pre-alignment filtering inside HMC cube. And what we did, basically, is that we observed that the data movement from CPU, from main memory to the FPGA or to the GPU to implement Gatekeeper, Shoji, all this work is very expensive. So we said, okay, we will utilize these technologies without using any FPGA or GPU links to the main memory. So we would like to implement something like that. These are the DRAMs, and this is the logic layer. And we said, okay, we studied the properties of this hardware. That's why you really need to know what hardware you have in hand, such that you design the algorithm first, then you implement it in the hardware. But don't pick ready-made algorithm and you try to match it to existing architecture. You are not leveraging the full power of the hardware. You really need to know how you can achieve the best or the less execution time and the best memory bandwidth out of this queue. So the best thing to do, in each DRAM we have banks. In each bank you can do memory access. And to access all of these in parallel, you need to divide your data to be each piece of the data in single bank in this layer. So each layer, if your data is, for example, hash table, the index, then you need to divide it, and each division or each chunk of this hash table, you place it in single bank in a layer. So piece here, piece there, piece there, piece there, and so on. But you can't place it here and here, for example, because this will be accessed once. So you place all of them here in parallel such that you access the entire hash table in parallel. And then you do the computation at the logic layer, where you can access all of these data and you do some processing in the logic layer. But you could do and piece here, but they are totally different for different hash table, for example. Or the same hash table but different reads, such that you can do this as a single computation and this another computation, with a third computation, and so on. So you can process many of these texts together in parallel in this queue. So based on this information, we decided to redesign the hash table. The hash table by itself is huge, and we cannot fit it here, because to leverage the full bandwidth of this memory, our hash table should be implemented in bitwise, meaning that 0, 1, 0, 1, 0, 1, and so on. Why is that? Because we want to reduce the size of the hash table. If we store the seed, which each character can be represented as integer of 16 bits or 32 bits, imagine we have 12 characters per seed, and each location is integer or 64 bits. So imagine how many locations per seed you have. If you implement all of this information for many of these seeds, and here you are wasting the resources. So we said, okay, let's do this kind of hash table. So I'm getting the reference genome, and I'm splitting it into bins, where each bin is certain number of characters, but they are overlapped, such as they don't reduce sensitivity at the boundaries of these bins. So how I build the hash table here? So I said, let's pick a seed of size 5 characters. And let's enumerate all type of values that I can get out of text of size 5. So it can be all of it A's, or I change this into C or G or T, all the way until all of it T's. This I do it for all bins. And then I pick the bin here, and whenever I find this seed, I record 1. If I find this seed, I record 1. If I don't find this seed here in this bin, I record 0. And I keep doing this for the entire bin. Then I have another one for the next bin. So I have 2 bins here for this bin and this bin. And I keep doing this for all of these, and what I actually store is this bit vector. I don't store this, I don't store the location or anything else. So I just store this with the index of the bin. And by doing that, my hash table or the index structure is really minimal. It's very small, where I store just bits. And what's the length of this bit vector? Because you are smart people in statistics, so you know that I'm enumerating all of these, and each character having 4 possible values, either A or C or G or T. So 4 times 4 times 4 times 4 times 4, which is 4 to the power 5. That will give you the length of my bit vector. And I store how many of these depends on the length I choose here, or depends on the overlapping between the bands and the length of your reference genome. If you compare this size to the reference genome, it's really small, to the hash table, for example. Okay, and this is the equation for the length or for the memory size you need. And this is what we choose as parameter, but again, you can choose these parameters based on your needs. And this is how we do mapping based on our modified version of the index. So we pick the read. Now we already create the index. That was the index. And this is the read. And exactly as we do in Yellow Pages, we pick the seeds from the read the same way we did for the index. So we get the tokens from the read, which is size of 5, We get all possible tokens that we could get from the read, and then we convert it to the bit vector, same method as the hash table. So whenever this exists, we record 1, otherwise 0, and we got this bin for read. And we already have a collection of bins for reference. And now the time to compare this to the bins we have for the reference genome, which is bitwise to bitwise operation. And whenever we get the match that this seed exists in this bin and in the bin that we have the reference genome, we record the 1. And then we accumulate all the 1s or all the shared k-mers between this bin and the other bin. And then we get the number. If it is less than threshold or more than threshold, we do something. Or either we send it to core again to do some processing, or if we are able to implement inside the logic layer of the cube dynamic programming algorithm, then we don't need to move the data to the CPU. But it was a huge amount of workload. This is a big project that we proposed. So what we didn't implement dynamic programming inside the logic layer. So we still need to move, after pre-alignment filtering inside the cube, we still need to move the correct answer to the core such that we do dynamic programming there, or needle man launch. And this is the full workflow for the read mapping. Yeah, same idea. I'm going to skip this. And you can notice that there are simple operations, just comparison between text and record the bitwise operation. Then you compare how many ones, or you count them first, how many ones, then you compare them to a threshold. That's it. It's very simple operation such that you can fit it many cores here in the logic layer. And it's highly parallel because we split the hash table into many cubes, many cores, and all of them work in parallel with different banks, and then you have the core inside the logic layer. And it's memory bounded because at the end you need this many of bits for each bin, and you have large number of bins, or depends on the reference genome you have, if it is a human or virus or bacteria. So at the end you need to utilize your memory wisely such that you can fit more cores inside the cube. And this is what we implement inside the logic layer, which is the computation step inside lookup table. So the first part was about splitting the hash table and the bins, and this is about the computation. So what you really need to implement as a computation core is the comparator, where you compare the seed stored from the bin of the read and the seed stored in the bin of the reference genome. You just need to compare them and record how many matches you get out of this. And then you accumulate the result and you increment it by 1 every time you see you got a match. And whenever you see the threshold, then you said this is a good guy, keep it and send it to the dynamic programming algorithm. So it's very simple computations. There's no here complex algorithm behind it. These are the details of the implementation, so I'm going to skip them. And this is the paper. We have the source code published as well, so you can go and check. Okay, so this is another paper published this year. What they did here, they did something about cache. So I didn't talk about cache, but as I mentioned in the previous slides, where we have the main memory, you move the data to the core, where you have inside the core several caches, which are very fast memory, but very small size. That's why we cannot have either caches or main memory. We need both of them, such that you can move big data from the hard drive, but the processor cannot handle this big data. So we need to buffer it inside a buffer or waiting queue inside main memory, then cache, then CPU. And what they did, they said, okay, let's use the cache. Our work using the main memory. But here in this work, they're using the cache, which is the next step. And they said, they observed some of our own pre-alignment filters that they are good at certain configuration and not good in certain other configuration, such as the read length and edit distance threshold. And then they said, okay, let's pick some of our own work for this specific read length or this range of edit distance threshold, and some other words for this kind of a configuration. And they place all of these pre-alignment filters together. So they have many pre-alignment filters working together, and all of them implemented inside cache. So the problem in such scenario is the overhead. So you are not allowed by this simple computer or this conventional computer we have, you are not allowed to do any computation in main memory or in cache. You only can do computation inside the core. But you need more advanced things like the HMC cube for main memory where you can achieve computation. And we don't have such capability for cache. No company proposed any hardware that enables you to do processing inside cache. So they proposed their own chip that they couldn't implement, of course, because this is academic research, but they increased the overhead of the cache. So here we have the DRAM, here we have the cache, single level or 3 levels of caches, and then you have the CPU. Oh, sorry, the people there won't see. So maybe we can move to the previous slide and explain it. Yes, this one. So I have the DRAM, I have the caches, and they have the CPU core. They implemented inside this cache, but this cache has certain size and a certain power budget, energy budget, and so on. So they increased the size of this by too much. And how much? The size overhead is around 17% or 16.4% of increase. So this might be not that bad, but the industry people always question why you need this kind of overhead. They might need to increase or change some other parts of the computer because of the cache getting bigger. Maybe you are exceeding the power budget, then they will have some thermal issues and so on, some constraint to be violated. But this was a good try, where they have this different pre-alignment filtering working in parallel, all of them working in parallel, depends on your configuration, the distant threshold, the read length, and so on. And here, they said by using the cache only with single pre-alignment filter, and this is the size of the cache, they could achieve this much of speed up or throughput. And by doing many of those pre-alignment filtering together inside the cache, they can achieve this much of throughput, where they can process many of these strings in parallel. Okay, so now we cover many directions, many research efforts for the last 10, 15 years, where we process some seed filtering very fast, we do pre-alignment filtering, different technology, very fast. Now how about dynamic programming algorithm? So as I told you, all the existing work, either they compromise the accuracy and they achieve less than quadratic time. So for example, one of the latest work, rather than n squared, we could achieve n to the power 1.6, which is a great improvement, but they are compromising the accuracy of the dynamic programming algorithm. So you could do that, or you could accelerate it using hardware where you have many lookup tables or many cores, and each one of them process 2 pair or 1 pair of strings, for example. So you can filter or examine or verify many strings in parallel at the same time. So one of the recent paper called Darwin, where they achieved 15,000x of speed up by doing specialization to the dynamic programming algorithm. And what is Darwin about is, so I'm going to skip this, these are details. They first, of course, analyze the execution time, the profile time for the dynamic programming algorithm, and they said, okay, to use your own CPU, your own computer, we need this much of resources, this much of ALU, arithmetic logic unit, inside the CPU, and this much of load and store from memory. And we need this much of cycles, CPU cycles, so the CPU cycles in processing core much faster than FPGA cycles, for example. But the best thing in FPGA, you can do many things in parallel. In CPU, you cannot. Everything is sequential. Even if it is 3 gigahertz or 4 gigahertz, everything is sequential. So they need this much of cycles and this much of energy spent on computing this dynamic programming table in your own computer. However, in hardware accelerated, in this technology node, and forget about it, just details of the technology, they need only single cycle to achieve this, and this much of energy and this much of energy for the logic only of the communication. So it's really cheap to achieve in hardware. But what they did is that they divide the dynamic programming tables into pieces. So this is a piece, this is another piece, another piece, another piece, another piece. And then they implement every single piece using systolic array. Now the question is, I told you there are data dependencies between the cells of the dynamic programming table. Now how they could achieve this division? Think about this line, for example, this row of this submatrix. How they could compute this cell without knowing the value of that cell, for example. That's a question I cannot answer. Honestly, myself, I didn't understand it from the work. So it could be a reason how they couldn't compromise the accuracy. But in my opinion, if they do this method, then they are compromising the accuracy to accelerate the dynamic programming table. And this is the only way to go if you want to parallelize the algorithm itself. So they need this much for the implementation. This is what size of systolic array and what size of memory they need. And they implement this for customized chip. It's not FPGAs, not GPUs, not SIMD, and so on. So they use this target technology. Now, this is simulation work, of course. They didn't fabricate it in the company. But they have FPGA results just as a proof of concept, but the 15,000x of speed up coming from customized chip. This is the speed up they get compared to idlib, which one we use to accelerate in Gatekeeper, Sneak Snake, and so on, where they achieve 5,000x of speed up. So this Darwin is not done by our group. This is from Stanford University. And what we did, we said, okay, we already have some experience in this HMCQ, the 3D stacked memory, where we have several levels of DRAMs. And we have good experience in doing that for gram filter, for example. Then we said, okay, why not doing dynamic programming inside the HMCQ to alleviate the need for moving the data from HMCQ to the processor, for example. So we want to do gram filter and dynamic programming algorithm, for example, inside. And this is what we call bitmap. So this is our recent work where we submitted just last month, and we're waiting the results, so it's under review. But we will be happy to share the paper with you if you like. And we achieve more speed up than Darwin without compromising the accuracy. So we are achieving the full dynamic programming, but we are doing many of them in parallel inside the HMC cube without data movement. So it's very energy efficient compared to processing dynamic programming and processes. Okay, so this is the conclusion for the three key directions. Read alignment can be substantially accelerated using computationally inexpensive and accurate pre-alignment filtering, and I present many of them from our group and outside our group as a recent work. And we would like really to have fast and accurate pre-alignment filtering designed for specialized hardware. And accurate pre-alignment doesn't mean 100% accurate, but at least we need 0 false negative. It's okay to have false positive. All the three directions are used by mappers today, such as Minimap2, BWMM, and so on. But they use different heuristics, different algorithms, and we would like to have the best out of all of this work integrated into a new mapper, which we are working on these days. So pre-alignment filtering does not sacrifice any of the aligner capabilities. So by using heuristic at the pre-alignment filtering without any false negative, you are always on safe side. You are not compromising anything coming from the dynamic programming table. So you can still get the correct result in your BAM file or in your SAM file. You got the correct CGAR string, alignment score, different scoring system for protein, for DNA, for RNA, that will not be affected, as we are not modifying or replacing the verification step. We still have all of them. Okay, so now what's the future of read mapping? What are the future opportunities we have? Now think with me, if we have these sequencing machines that producing the read to us, could be short or long, could be accurate, inaccurate. Think with me that we got a very long read, as long as your genome. single piece, complete sequence of your genome. And 100% accurate. There's no sequencing error, just the SNPs or the genetic variation there. What will happen to read mapping? This sounds like very ambitious. We might not have it within the next 10, 20, 30 years, but we might have it. We don't know. So think about read mapping problem. All these pre-alignment filtering we design, all those acceleration work for dynamic programming table. What will happen to all of these? Should we just trash them? Will the area of bioinformatics will be dead, as some people saying? Okay, you need to think twice, actually. Think about metagenomics, pangenomics, and many other research fields, where you are comparing a complete genome to a complete genome. There is no read to genome comparison here. When you compare metagenomics, you are comparing a species to database of species, because you don't know what species you have, right? So you are always comparing genome to genome. And to compare them, you cannot use Hamming distance, for example, because you would like to know what variation you have between you and another individual, even if human to human. And some of the disease cause insertion, deletion, substitution, so you still need dynamic programming algorithm to compare the genome to genome. So to do that, and there will be some false positive when you compare virus to human, right? So it's very expensive to use dynamic programming table, and you still need pre-alignment filtering to know which species is closer to this in a very fast way, and then you use dynamic programming table to give you the exact differences and their location. So all the entire area of read mapping or bioinformatics or genome analysis, you still use read mapping, even if these sequencing machine getting better and better. And that will show you the importance of doing sequencing per population. It's not bad to have a single reference for the entire world, but it's not representative. For each country, for each population, for each group of people, we need to have our own reference. And this paper showing the need, and many other papers as well, showing the need for having such references. So as long as we are having the motivation to sequence more individuals, we will still use read mapping, which will be a bottleneck in any pipeline we are using. Okay, towards the end of the talk, did we achieve our goal? Did we have this smartwatch analyzing our DNA? Unfortunately not, and we need the help from industry. We are an academic institute, so we can't do everything by ourselves. So we still need the industry to jump in and start producing these customized chip for genome analysis. As the many examples I showed about Tesla and Preferred Networks, all these companies, they produce their own customized hardware for specific applications. And we need so for the read mapping or genome analysis. And we cannot answer how and where to enable the computation of genome analysis inside RAM, inside cache, inside the processing core with large number of cores, or inside the sequencing machine itself. And this is still open question for research. Where to place the sequencing machine. And by this, I come to the conclusion. So system design for bioinformatics is a critical problem that can affect even the society. It's not about wet lab and what you do inside university. This lecture is about accelerating key step in bioinformatics, which is a very initial step used in any pipeline where you compare some genetic data to another data. It could be RNA to RNA, DNA, protein, and all those involve some comparison with some data structure. So many bottlenecks exist in accessing and manipulating a huge amount of genomic data during analysis. And we cover various of these ideas. Some of them we were working on 2006, 2009, and some of them just get published. And this journey starts with my professor, who would like to acknowledge, John Alekan from Bilkent University, where I did my PhD, and my current professor, Onur Mutlu from ETH, and all the collaborators we've been working on all these nice projects, and the funding agency as well. And you can find all these projects and even the future ones in this link. And please don't forget that we are hiring students, so feel free to contact me, contact Professor Onur Mutlu, and as well as you can submit the application directly. I have these two references for you if you'd like to know more about other algorithms, other techniques they use in the past and currently and the future. So you can read these two nice, very nice references. I highly recommend them to you. And thank you so much. If you have any questions.
Okay, any questions or comments? I have some comments on the last slide of the two books. And the one book on the left side is a book that I translated into Japanese.
Oh, wow. Wow.
And on the right side in the book, I think in the section 9, my algorithm is inside.
Oh, wow. Great, great.
But anyway, yeah, it's just a comment. But anyway,
Great to know that.
I have some questions about the pair entries. So I think most mappers doesn't utilize any information of pair entries as two is nearby. Do you think it's possible to use such kind of information to speed up mapping?
Yes. So pair and read, right?
Yeah.
So these are like duplicated or two read alignment, right? So you're using dynamic programming here and another dynamic programming here, but you could leverage some information between them. So by accelerating the dynamic programming itself at this part, so you can do very fast computation for pair and reads.
Okay. Another question is that I think the FPGA has become larger and larger and faster, and moreover, it becomes easier to configure.
Yes.
And I think it will continue in the future. So, do you think there's any kind of more accelerations by using the future FPGA that cannot be done in current FPGA? Do you think?
So it can be done. So we are using Vertex 7 from Xilinx, PC709. There are more advanced one called Ultra Scale. So by using these, you can achieve very high speed up. Of course, the recent one, the more advanced one, where you have the HMCQ I mentioned next to the FPGA inside one board coming from Micron. So using this more advanced, not Ultra Scale, the more advanced one, you could utilize very high memory bandwidth next to the FPGA board without the need for CPU maybe. So if your virus or bacteria genome can fit inside the 3D stacked memory, you could do the processing within this chip without the need for a computer. And this is what we are looking for, actually. This is what we will investigate next.
How about multiple alignment? Do you think? How about multiple alignment? Do you think?
So if you'd like to align multiple
align
species or many reads at the same time.
Reads on more than 3 individual genomes.
Exactly. So this problem has its own challenges. It's different from single read alignment. But at the end, you'll still use dynamic programming table or some kind of indexing at least. And you could apply some of the method, but it will be great again to customize the algorithm and the hardware for each problem rather than using one general solution for all. So we could accelerate that as well by having the hash table in 3D stacked memories or cache, or even for traditional computers. These traditional computers still have very high power of computing. We could still leverage them to do the computation without the need for hardware, but if we design the algorithm to be smart enough, as one trick, for example, if you can avoid I/O as much as possible, rather than storing, saving the file, keep doing that to the hard drive. So if you can load some chunk of data, keep it in memory, process it using the CPU, then you are achieving very optimized computation. And you could achieve Snake, for example, as very lightweight computation. So you could do that for many species, even without the need for doing multiple alignments at the same time. You still can do individual one, then you combine the result, for example. Something like that of this direction. But of course, researchers need to investigate that.
Thanks. Any other questions, comments? Well,
Yeah.
so please.
Okay, thanks for the nice
Yeah.
alignments and also providing solutions on the hardware. So I have a question though, because there are lots of informations, for example, genetic variants also providing the read. So how about this kind of information
I see. So this is a very great direction. We were discussing this with Rican people and with the University of Tokyo, other campuses. So they would like something that you could leverage sequencing error profile, for example, or structure variation while you are doing read mapping. So they would like something to do this inside read mapping process. So this would be very nice to leverage all this information. But the good thing about all this read mapping, you keep all the structure variation, all these differences, you are keeping them. And you are still using dynamic programming to infer these variation for you. So you still have all the information on insertion, deletion, substitution, and so on. But it will be really nice if we can combine, for example, pre-alignment filter with a very fast aligner, the same component, same algorithm, such that, for example, the matrix, you already build it for a gatekeeper, for sneak. Do you really need to build another one for the dynamic programming table? Maybe you can convert the information in that matrix to that one. Those are also areas to be investigated. For example, Gatekeeper. Do you really need to do AND operation for all those bit vector? What if you do just AND operation for some of them and the other some together, such that you can avoid false positives? All of these need to be investigated actually to improve this work. How about sequencing error, for example? Some of the machine tell you that I'm having this profile of sequencing error. For example, Mina, they publish their own profile. So they tell you the first 30 bases or so, they are mostly erroneous. You cannot trust them. So you can leverage this piece of information inside your read mapper such that you provide a good information rather than this is a structure variation or sequencing error simply, and you can correct it. And we actually publish some of the works doing assembly polishing called Apollo. This is our recent paper. I'm going to have some slide for it. This is the paper where we do polishing based on the sequencing error profile and read mapping together. So we map the short and the long reads together based on the assembly, and then we do some heuristics, some machine learning model to polish the assembly based on the sequencing errors. So I have some details about it. This is the model we built, where each node is the character, and those are the overlapped reads, the mapped short and long reads together, overlap. And then we do a training algorithm. These are the costs we are paying for each node. So we assign some cost, and then the network try to train itself such that can infer whether this character should be A or C or G or T based on the choices we made over all these reads. So this is the training algorithm. This is the training algorithm we use. We could spend some time explaining it. Then we use Viterbi algorithm to infer based on the calculation we use for the training to clean the assembly from this to that. This is a very recent paper called Apollo, and the very nice thing about Apollo is that it's scalable. So you can handle plant genome, animal genome, which other tools cannot simply. They require more than 2 terabytes of RAM, which you don't have in this machine or even in clusters, and it will be crash soon. So this is the first good thing. The other good thing is that this tool can handle both short and long reads in the same run, in the same tool. You don't need to rerun your tool for short, then you get the result, then you do remapping for long, then you combine the result together. Does that answer your question?
One thing that people have, so they have released assembly of reference genome in place more genetic variants inside of reference genome. So we are not going to allow it to the latest reference genome.
Oh, okay. So you are asking if we should change the reference and map to another reference.
Yeah, so what's the common version?
Okay, so for this work, it's independent of the reference. Whatever you use, 37, 38, it's independent of what reference you use. But again, that remind me of a very new tool that we designed called Airlift. We just published the code over archive yesterday. And for the nice thing about this tool imagine you have a data, you have the reads, you have the reference. You already map all of these reads to the reference, right? And now, next 6 months, NCBI provide a new version of the reference genome. Now, do you really need to map again all the reads to the new reference? So we did a new tool such that you can leverage some information of the previous mapping and the new reference, where we can know the regions where the reference get updated, and we only do mapping for these new regions. And then we upgrade the entire BAM file from the old mapping to the new reference without remapping everything. And we were discussing for some collaboration here in the University of Tokyo. They were collaborating with the government to sequence genomes of Japanese people. And they already did this for short and long reads for the old reference genome. And they were looking for a tool to do that for the new reference. And we were lucky enough to publish this before coming here, such that we could provide them the paper and tell them, okay, this is our source code, use it please for the Japanese genome, where you can upgrade everything to the new reference without the need to repeat this, not for a single individual, but for thousands of them. Think about animals, plants as well, all other species. So the paper called Airlift. I can find it here. Screen is really big. Yeah, this is the source code, and you will see the paper somewhere here.
Any other questions, comments? Okay, so let's thank the speaker again. Thank you very much.
Thank you so much for inviting me.